In this lecture, we will cover the taxonomy, the microbial cell metabolism, as well as genetics of various microbial organisms. The role of a clinical microbiology department is to assist the healthcare provider with the diagnosis and management of any sort of infectious disease. Now, we do this by various tests that we perform within the lab, and that will include things like microscopic stains, cultures, the phenotypical characteristics of the organisms themselves, immunochemical testing characteristics, as well as molecular analysis and the antimicrobial susceptibility testing. Now, we use all of these in combination to help the healthcare provider to treat these various diseases based off of these different tests. Now, when we start talking about a clinical approach to diagnosis of various infectious disease, we kind of have to start to realize exactly what is the process that we go through to figure out and diagnose the culprit of the infectious disease. So to do that, we have to kind of learn what those particular microorganisms are, what they do, what their characteristics are, because we're going to use all these characteristics to help identify exactly what it is. So those characteristics that we're looking at, we call their phenotypical characteristics. We can also look at the genetic characteristics, but that's kind of that newer development in technology that is helping us to look at the genetic characteristics. So when we talk about genetic, we're talking about the genetic homology of that particular organism. And when we look at that, we're looking at things like the DNA base composition and ratio. And so when we do that, we're comparing the cytosine and guanine content against that total base content, and we're using that as an indicator of the relatedness. And so we can also use the nucleic acid sequence to analyze that particular organism, and then we use it actually uses the order of those particular bases, so those base pairs of those various carbohydrates, along with the DNA or the RNA sequence in order to determine what's similar. Now, just because an organism may test out to be one thing, it may not necessarily fit those phenotypical characteristics. So when we start doing the DNA and the nucleic acid type of testing and looking at that RNA, sometimes you may find that an organism has kind of adapted over time, and so something has changed in its phenotypical type of presentation, and so then these organisms might get reclassified. So this is where you kind of hear what you're going to be most familiar with will be like coronavirus, how things have kind of changed along the way in its genetic makeup, and so its characteristics for its DNA makeup are starting to change a little bit, and how it presents in a clinical setting changes a little bit. So it's still in that same family, but it does have some genetic differences from where it originally started. So if you think of it as two individuals having a child, and then the offspring of that child produces some different genetic characteristics based off of different things, environment, things like that. Obviously, what we've learned over the past several lectures is your environment can actually influence your DNA and genetic makeup and make alterations that can genetically be passed down. So you kind of got to think of it in that environment. But going back to those phenotypical characteristics, so when we talk about the phenotypical characteristics, we're talking about how does that particular organism live, what makes up that organism, what kind of features does it have. So we're looking at things like its macroscopic morphology. So this is going to be what it looks like. How does it appear when we visualize it with our eye? So we're going to look at things like size, texture of how it grows on that plate, the color of it, does that colony raise up, is it sunken in? So we're going to talk about some of those things as we kind of progress through our lectures and through this course. We also look at things at the microscopic characteristics. How does that organism stain? What size is it? What shape is it? What does it look like in a microscopic environment? We look at those staining characteristics. Is it a gram-positive or gram-negative? Is it acid-fast? We also look at the environment that it needs to grow. Does it have an optimal temperature that it grows at? Does it have specific oxygen need? Must it grow in CO2? Does it have to be grown in a particular pH? Those growth characteristics are going to help you identify that organism, as well as those nutritional requirements. What does it need to grow? Does it need something a little extra in order to help it grow? The biochemical characteristics. So when we do some different biochemical tests, we want to see how does it react in that test. Does it use glucose a particular way? Does it use other sugars instead of glucose? We look at antigenic properties. So this is going to be the serological type testing. So we're looking at how the antigens on that particular cell responds in different environments, and in those tests that we can do to test for particular antigens. We also look at resistance profiles. So we look at different chemicals and antibiotics to determine what kind of characteristics that particular cell has to be able to grow in that environment or not grow in that environment. So again, we kind of take all of these things together, and it's like piecing the puzzle together. Once you take all of these different characteristics, you can kind of narrow down what particular organism it is based off these characteristics. Taxonomy is the scientific study of and the classification process. So with any organism, it can fall into this classification scheme, which is divided out with different categories. So first you start with life, and then you move down to domain, which has 3 categories: the bacteria, archaea, and eukarya. And then after that, it's divided into kingdom, and then that's divided into phylums, class, order, and family. So within a family, the organisms have very common attributes. Then it's further divided into genus, and one genus can have several species within that genus. And then the species, of course, is the most basic taxonomic group. So here we do have that chart that we looked at in intro. So it kind of further divides out those 3 main domains into those further kingdoms and further down even below that. So once we kind of talk about some of these, we're going to kind of talk about some of the different organisms that are within these various divisions. With binomial nomenclature, we break the organism down to the more simpler form, which is the genus and the species. And so with this, the first letter of the genus is capitalized, but it can be abbreviated. So for an example, with Staphylococcus bacteria, you could abbreviate it with just an S period. Species names in lowercase, they're in lowercase, but they're never abbreviated. So for instance, pneumoniae would be spelled out either S. pneumoniae or Streptococcus pneumoniae. And so with this, both are italicized. So if you're typing it out, you would italicize it. But of course, if you're writing it handwritten, then it would be underlined to indicate that it is the genus and the species. So you will use this throughout your microbiology course, so you need to be very familiar with how you write out an organism's name with the genus and the species. Microorganisms can be divided into prokaryotes, which are organisms that do not contain a nucleus or any other membrane-bound organelles such as mitochondria. Or they can be divided into eukaryotes, which do have a nucleus within the cell. All right, so we know that when we're talking about organisms in microbiology, we're talking about your fungi, your bacteria, your parasites, your viruses. Now, for the purposes of this lecture, we're just going to kind of start finishing out focusing on the bacteria. Most of what we're going to cover in micro will be bacteria. It's a real in-depth category, so we're going to kind of focus on those phenotypical characteristics of the bacteria. Once we actually start talking about those other organisms, we'll talk about those phenotypical characteristics within those lectures, but now we're just going to kind of shift gears and focus mostly on our bacteria. Now, when we're looking at organisms and talking about those phenotypical characteristics, one of the ones that we talk about is how it appears. So we talk about the macroscopic appearance as well as the microscopic appearance. Now, when we talk about microscopic, we want to talk about the shape as well as the staining characteristics. Now, Talking about shape, we can kind of divide that into a few different forms. One of the first forms that we talk about is coccus. Coccus is the singular form of this terminology, and it just means that it's a spherical type of form or a rounded type of form. If we talk about a plural form, it would be cocci. So if we talk about two or more, we're going to say cocci. If we're talking about just one, it's coccus. So something like Streptococcus pneumoniae is talking about one genus of Streptococcus. If we talk about Streptococci, we talk about the various groups of Streptococci that are in that particular genus. All right, so if we talk about Diplococci, we're talking about a pair. So we can see our coccus in pairs, or cocci in pairs, so that will be referred to as a Diplococci. So you can kind of see here where the organisms are kind of joined up, just sitting kind of side by side. With your Streptococcus pneumoniae, so it does do something a little different where it kind of pinches at the end. So they're still rounded, but they're more of an oval shape. And then your Neisseria, you can see where it kind of joins together, where it kind of has that flattened edge. And you'll learn more about that visual appearance once we actually talk about those organisms. But for now, just know that you can have Diplococci or pairs for your organisms. You can have chains. So this will be an example of your Streptococci. These organisms will microscopic feature will be in chains. And then we also have clusters. Your Staphylococci tend to be in clusters. So if you think about like a cluster of grapes where they're just all kind of booted up there together in just a little cluster. We also have bacilli, and so bacilli would be your plural form for this terminology, bacilli, or bacillus. So if we're talking about one organism, we're talking about an organism that is in the shape of the rod. Now, the facility that you work at may still use the term rod. Some of them will use bacilli. So whatever terminology your facility uses, that's what you will use to report it out. Now they can vary in size. All of these organisms can vary in size. So just because it's a rod, it doesn't mean it's going to have one size fits all. You will have some that are a little bit more plumper. So you can see where this one looks a little wider. You can have some that are shorter, longer. You can see where this one's a little thinner, and then this one's kind of in between. You can have filamentous type of looking organisms, so there will just be like a long filament. You can have organisms that will actually arrange, or bacilli that will arrange in chains. You can have some that are more curved, so you can see this kind of curved shape to this organism. You can have coccobacilli, and so it's going to have that more elongated shape. You can see your Streptococcus up here is more rounded, more that egg type shape, where your coccobacillus is more elongated, more a cigar type of shape. You can have a fusiform. So with this, you've got those pinched ends where you kind of looks like a spindle sort of. And then you have spirochetes. So your spirochetes are rounded or curved like a corkscrew. And this is going to be an example of this would be like the organism that causes Lyme. That is a spirochete. Or the organism that causes syphilis. That is a spirochete. So now we're going to talk a little bit about the various bacterial components. And so we can divide this up into the outer cell structure, where it has its actual appendages and various things depending on the particular organism. And so we refer to this as the cellular envelope. And then you also have all of the components that are on the internal part of the cell, or the cytoplasm or cytosol. Now, depending on the particular organism that it is, the outside cellular envelope can vary from organism to organism, but you basically have kind of the same pretty much overall composition. So you have the cytoplasmic membrane as well as the cell wall. Now, It can vary depending on whether whatever particular bacteria it is. And then you can also have appendages. So you can have things that are kind of sticking out from that particular organism or things that are coating it. So you can have things like villi. These are going to help with the motility of the organism. So if you have organisms that are able to kind of move around, you're going to have these little things on the outside edge that are going to help it to move. It could have a capsule or some sort of slime layer on that outside edge. And what that's going to do is just kind of help it with some of its various virulence factors and its protection. It's sort of like its armor that it uses to protect that particular organism. And you can also have some that are within they have endospores, so they have these little spores within inside of that particular organism. Now, if we talk about what's inside of the organism, the cytoplasm or the cytosol, we're talking about all the things that are inside of it. So once it's inside of that organism, it can contain things like the ribosomes, the plasmids, the DNA chromosomes that are within it, the messenger RNA, the various proteins and metabolites. And so these are all things that are going to kind of give it those general characteristics. If you're breaking down the DNA or you're looking at that messenger RNA that's within that cell, you're talking about everything that kind of makes up that cell, that DNA. So all that is housed on the internal structure of that cell. How it responds, the various proteins and things that it produces, how it responds to its environment, all that takes place on the inside. So when we do some of these tests, it's based off of what's happening inside of that particular cell or based off of the composition of that cell wall or that cell envelope. Now, one of the first characteristics that we note with organisms when we look at it at a microscopic level is that cell wall composition. Now, the cell wall of the organisms do contain a moraine layer or a peptidoglycan layer. And this outside layer of the cell is what's going to determine how it actually stains and how it absorbs that stains and retains that stains. And it's got to do with the various layers of that particular cell wall composition, and that's what differentiates the different types of organism and how we first kind of start to classify them into a particular category. Now we'll have some videos that kind of helps give you a better depiction of this actual cell wall. But here in this image, just going to kind of break it down for you. Everything here would be the inside of the cell. So we're kind of looking at all of this as that outside of that cell wall that just kind of goes on to the outside of the environment of that cell. So here, if the organism has any sort of appendages coming out, any sort of flagella, anything that's going to kind of stick through that cell wall, and that's what's going to kind of help it to move around in its environment. And it also, but depending on the particular organism, it could have this little sexual type of appendage here, so it actually uses it to transfer its DNA makeup or material, that coating and everything. And then from there, depending on the particular organism, you could have a capsule. There could be something coating it, slime layer, various things that could be on this outside. And then depending on what particular organism it is, so if it's a gram-negative, you actually have this outer membrane here. So you have this layer that contains this lipopolysaccharide layer on the outside of that cell wall. And then depending on the particular organism, of course, you have this little layer here, this moraine layer here. As you can see in the gram-positive, it's very thick. You got lots of layers to it, whereas in this gram-negative, it's actually in between this outer membrane and then the cytoplasmic membrane. And so you do have a little bit of a thinner layer of that actual peptidoglycan. Now, because you have this thinner layer, that will actually affect how it stains. And so we're going to kind of talk about that reasoning behind that stain. But then you kind of keep going in, and then you got the cytoplasmic membrane that continues to make up the cell wall. and within that particular membrane, it's got different things that actually make it up. And of course, that cytoplasmic membrane is what's going to kind of be responsible for, that permeability of anything that's inside of the cell actually passing through. And so you can kind of see that here, where you've kind of got something that's just kind of passing through from inside that cell to the external. So that's the particular layer that it will kind of work its way through to get to that external part of that cell. As far as when it's acting on the host and its responses to the host, all of that's going to come through that particular cytoplasmic membrane and outside of that cell. And here we have another image that just kind of depicts the differences between that gram-positive and that gram-negative cell wall. So as you can see with the gram-positive, you've got that thicker peptidoglycan layer, which is made up of some different things within it. So it's a thicker layer. Obviously, if it's a thicker layer, it's going to retain that stain a little bit better than a thinner layer. And so we're going to kind of talk about a little bit later of the various stains and how it acts upon these various layers. But you can see with your gram-negative, you've got that thinner layer there, and then you can see that cellular structure where you have that that whole cell envelope encompasses that cell membrane, is cytoplasmic membrane, as well as that cell wall. So you can see the differences between the two of the gram-positive versus the gram-negative. So obviously you can see the difference in that structure is going to stain a little bit different just because it is made up different. And this is what causes us to first kind of differentiate the cells. So we're going to look at the shape of it. Is it cocci versus rod or one of the other shapes? And does it stain gram-positive or gram-negative? Or it could actually have a particular wall that will not retain either of those stains, and it's what we would call like an acid-fast type of staining. So again, it's just got to do with the makeup of that particular organism in the wall and how it absorbs and interacts with those various stains. Now with bacterial cellular metabolism, we do know that an organism must go through various processes in order for that organism to survive and actually replicate. Now, part of that process is metabolism of the various nutrients in its environment in order to create energy, in order to survive and replicate. Now, its various metabolic processes are actually some things that we can look for to help with identification of this organism, and we do that looking at the basic phenotypical type of biochemical reactions to help us understand what that particular organism is based off of how it interacts with its environment. Now, the nutrients for an organism do enter through a couple of different processes. It can enter through diffusion, in which it actually just kind of diffuses across that plasma membrane. Or it can happen through an active transport, in which there is some sort of carrier molecule that will actually transport that particular nutrients inside of that cell. Now, once an organism has those nutrients and it's actually starting that process to make that energy to do that process of replication and surviving in that environment, it uses a couple of different pathways to actually generate that energy. Now, one of those pathways is the Enden-Meyer-Parnas pathway, Enden-Meyer-Hoff-Parnas pathway, which is a glycolysis type of pathway. And it is the major pathway that most of the organisms that we're going to discuss will actually use. Now, this pathway does not require the use of oxygen, and it will ultimately end with a generation of an ATP. Now, the tricarboxylic acid cycle, otherwise known as the Krebs cycle, this is an aerobic oxidation of pyruvic acid. There's also some alternative pathways that aren't used as often, but they are still used. These are the pentose phosphate pathway. And in this pathway, glucose is converted multiple times through various steps and processes, which ultimately will end up producing a pyruvic acid. We have the Enterner-Dodorov pathway. This is where you have glucose that's converted to pyruvic acid, as well as a glycosaminoglyceraldehyde 6-phosphate, which will actually enter into other pathways, and then it can assume one of the other pathways to generate that energy. And it is an aerobic type of pathway which requires oxygen, and it is actually used by bacteria that lack some sort of glycolic enzyme to be able to break that down. And ultimately what will happen is it will lead to the production of some sort of ethanol in its environment. So when it makes that energy, it's going to end up producing an ethanol. We also have oxidative phosphorylation, and this uses NAD plus and NADP plus to actually generate an ATP molecule from an ADP. You can also have fermentation and respiration. And respiration is an oxidative type of process. And this involves breaking down carbohydrates to glucose that is then converted to pyruvic acid. Now, in microbiology, another term that we use will be the type of respiration that an organism has. So if an organism is, if it cannot grow without the presence of oxygen, so in other words, it needs an oxygen-rich environment in order to grow, that is considered an aerobic organism. If it has to have that oxygen, it's an obligate. It's obligated to grow in the presence of oxygen, or it's a strict aerobe. So it must have oxygen in order to grow. Now, an obligate strict anaerobe, so ana meaning without, that anaerobic organism cannot tolerate any sort of oxygen. If an organism is facultative anaerobic, it can use an aerobic respiration or fermentative type of process in order to generate that energy. So it can actually do its metabolism a couple of different ways. In aerotolerant organisms, these are anaerobes that do not use oxygen but are not killed by small amounts if it is present in the environment. So they don't really need oxygen in order to do their metabolism process, but they can still grow and still function if there is a little bit of oxygen present in their environment. And then we have microaerobic, and this includes both capnophilic and microaerophilic organisms. So if it's capnophilic, then this means it requires an increased concentration of carbon dioxide. And then a microaerophilic means it just needs a reduced oxygen environment. So just needs a lower level of oxygen with a little bit slightly more elevated CO2 level. Now, part of the ways that bacteria do cause the disease process is the fact that they are able to release toxins. So bacteria can produce a couple of different toxins. You can have exotoxins and endotoxins. Your exotoxins are actually secreted by a living organism or a living cell. Typically going to be your gram-positive and gram-negative organisms. And it's usually kind of limited more to specific species. So something like diphtheria or cholera, those particular toxins that are produced by those live living organisms. You also have endotoxins. So an exo, you would think, yeah, it's outside of the cell, so it produces it and releases it while it's still alive. Whereas the endotoxins, these are bacterial toxins that are maintained inside of that cell wall that are released once that organism ruptures or pops open. So typically it's going to be some sort of cell death that causes that organism to pop open and then release the internal contents, those endotoxins that are inside of that cell. It's not species specific, so it can elicit the same effect regardless of the particular bacterial species that is present. Now again, your exotoxins are going to be live living organisms, whereas your endotoxins are going to release with the breaking of that cell wall of that organism. Now, as we are aware, anything that is living has some sort of hereditary type of information that is encoded within the nucleic acid. There are two major classes of nucleic acid. We have the DNA as well as the RNA. Now, RNA itself does play a very essential role in several of the genetic processes that take place in the prokaryotic and eukaryotic cells, and this is going to include the regulation and transfer of that particular genetic information. Now, as you know, the prokaryotic cells do not have any sort of nucleus within it, so the genetic makeup is not contained within a nucleus. But with your eukaryotic cells, it does have a true nucleus, and so that genetic material is maintained within that nuclear envelope. Now, when we look at genetics of a cell, we're looking at this to study the genes, the variations, and the hereditary makeup of that cell. And what that can do is that it can actually affect the organism's ability to multiply, survive, adapt, and cause disease based off of that genetic makeup. So when we're really trying to get to the bottom of a particular disease or try to figure out what's causing an infection, if we start seeing something that's a little bit different or doesn't follow the typical pattern, this genetic makeup is going to kind of help us to figure out exactly what's going on with that organism or try to actually diagnose whatever that disease process is. Now, we can do that through a couple of different methods that are used in micro as far as to look at the genetic makeup of an organism. And we do this by the hybridization methods as well as the PCR testing. Our PCR testing will help us not only to identify the organism, but as well, it will also tell us if that organism has the genetic ability to have any sort of antimicrobial resistance. Now, DNA does consist of deoxyribose sugars that are connected by a phosphodiester bond. And this base, and so we call that a base pair. When you've got those connections, that's a base pair. Those bases are covalently linked together. And then that actually will give the code, that those links together will give the code for the DNA molecule. Now, the four nitrogenous bases that are used include the two purines, adenine and guanine. As far as the base pairs, your base pairs are adenine that is paired with guanine. And then you also have a base pair with pyrimidine base that is containing a cysteine and a thymine. Now with the RNA, the difference is that you have a uracil that will actually replace that thymine instead. So you'll have that pairing up with the uracil instead of the thymine. Now, when you're actually performing any sort of molecular testing and you're looking at things at a genetic level, then what you're basically looking for is that pairing, how those pairings kind of fit together and what the sequence is of those particular pairings. And then particular sequences will translate over to a particular genetic makeup. And then when you kind of compare all those together, that helps give you the information that you need. Now, a DNA sequence will actually encode a specific product or an RNA or protein, and this is defined as a gene. Now, we do have thousands of genes in an organism that will actually encode messages and blueprints for the production of one or more proteins, and then the RNA products that play an essential role in that metabolic process within that cell. Now, each of the genes within that organism are going to contain that particular organism's genome. So the genome of an organism is going to include the particular chromosomes and the extra chromosomal mobile genetic elements that are within that cell. Now, in order for a cell to survive, then it must replicate. So bacteria will actually replicate by something called binary fission. And so this is a form of cell division. And what will happen is it will result in the production of two daughter cells from the one parent cell. As part of this process, the genome will actually replicate within each daughter cell, and each daughter cell will actually receive an identical copy of that functional DNA. Now, replication is a very complex process, and it is mediated by various enzymes such as DNA polymerase and cofactors that will actually, come into that cell and actually initiate that process. Now, that process is pretty complex. It goes through goes through a four-step type of process, and where there is first it starts off with an unwinding or a relaxation of the chromosome's supercoiled DNA. And then after that point, the separation of the complementary strands. So that DNA is uncoiled, then it is divided up into complementary strands of whatever the parental DNA is. And then each strand is going to serve as a template or a pattern for the synthesis of that new DNA strand. And then the synthesis of the new strand will actually start to take place, or that daughter strand will start to take place. And then from there, you're going to have a termination of that replication, and then you're going to have those two identical chromosomes that have been reproduced out of that one cell. And so then you'll have one for each daughter cell where that division has taken place. Now, gene expression is the process of information that's actually encoded in that genetic element. In other words, what's encoded in like the chromosomes, the plasmid, the transposons. And so this will actually result in the production of some sort of biochemically functional molecule that will include the RNA and various proteins. Now, the process of gene expression does take place in two steps. You have transcription and translation. In transcription, you actually have that DNA molecule or that DNA strand that's copied, and then it's transcribed to messenger RNA. Now with translation, that information that is provided from that messenger RNA is then converted to a specific amino acid sequence. So that whatever that messenger RNA is coding for, it's basically doing that translation so that and conversion within that cell. Now, organisms can occasionally go through something that we refer to as a genetic change. So, recombination happens when there is a segment of a foreign DNA that will actually combine with a homologous or an identical portion of the bacterial cell as part of that original chromosome. Now, what this will actually do is it will form a partial hybrid type of chromosome that will have segments that originate from both the donor as well as the recipient. Now, a segment of DNA moves from an original bacteria cell to another organism, and then that particular DNA is actually exchanged within that organism. Now, recombination results in a combination of a new gene, which can produce additional biochemical pathways and enable the organism to adapt to its environmental changes that are taking place around it. Now, another way that you can have some sort of genetic change is via a mutation. And so this is when there's an alteration in the original nucleotide sequence of a gene or the gene within an organism's genome. So this particular alteration can involve a single DNA base within that gene, or it can involve an entire gene or several of the genes. Now, mutational changes in the sequence may arise spontaneously, or it could just be by an error that was made during that DNA replication. Mutations can also happen because there's some sort of mutagen in the environment, and so this would be something like a chemical or physical factor, or some sort of biological factor that will affect that particular cell. And so these alterations in the DNA base sequence can result in a change in that base sequence of that messenger RNA when that transcription process takes place. Now, sometimes these mutations can affect the cell to the point that it could cause something to happen where that cell is no longer able to survive, or it could be something where it changes something observably in that phenotype of that organism, so how it interacts with its environment. Some of these changes could go unnoticed, or they could be noticeable, so it actually affects it to the point that it changes its phenotypical types of observations. An example would be maybe an organism has had a mutation to the point that it's able to survive and evolve its particular environment, so it is able to actually survive, or how it is affected by the whole process of elimination, or even mutations to the point that a particular antibiotic can't kill it anymore. Now, an organism can actually exchange information or genetic makeup. It can do this by exchanging that information with other organisms or a DNA from a particular donor cell. And so what it does is it will actually go through a process in which where it exchanges that information, and it can be through transformation, transduction, and conjugation. With transformation, so this actually involves the recipient cells to uptake of a naked or free DNA that is released into the environment when another cell, and we're going to refer to this as the donor cell, it actually dies and undergoes that lysis process where that those components are kind of free floating in that environment now. Now, that transformation of that genetic makeup material will actually be used within that cell, and it will do a recombination within that cell, and then ultimately it could do something like pull the genetic material of a bacteria that that cannot be killed by a particular bacteria, and then it will actually transfer that information to the recipient bacteria, who will recombine that within its DNA makeup to ultimately evade that particular treatment option. So in other words, it's no longer able to be killed by that particular antibiotic. With transduction, you have that DNA from two different bacteria that will combine together in one cell, and then you have that recombination process that will take place, and then you have a virus that is capable of actually infecting a bacteria. These are called bacteriophages that actually aid in this particular process. So part of this bacteriophage's life process is to actually integrate their DNA into the bacterial cell's chromosomes, and then you'll have that viral DNA replication and expression that will happen within that cell. And then ultimately what happens is it reproduces a newly formed recombinant viron, which is a viral particle, along with additional multiple virons, and then it's released when that particular bacterial cell will lyse open. With conjugation, this process involves a cell-to-cell contact and requires mobilization of the donor's bacterium's chromosomes and other mobile genetic elements. And so with this process, what will actually happen is the nature of the intercellular contact is not well characterized in all of the bacteria species that are capable of doing this process. However, we do know that in E. coli, the contact is mediated by a sexual phallus. So that organelle from that donor cell will actually establish a type of bridge that will transfer that DNA from the donor to the recipient cell.